to the lab.
Thank you. Oh, how
So
exciting.
what we're walking into right as you step into the lab over here is our molecular biology department. So at the start of what we do in our process is we take yeast and we program our yeast to be human. So we use advanced gene editing tools and we work with our yeast cells so that when we feed them all the food that they need and we ferment them, they produce human proteins. So all of that work is happening in this room, including high throughput screening of our yeast strains, which allows us to look at a lot of different ways we modify the yeast, looking at thousands of different clones of yeast to figure out who's the highest performer, which yeast strain is making the most amount of protein with the same structure and function as what we've been producing to date. So we have many QC steps and quality assurance programs to make sure that each advancement that we have in our yeast is going to yield exactly what we want. And all of that happens in this room.
That's great. And you want to increase the yield, obviously, right?
Absolutely. Yeah. The way that we improve accessibility of what we're making is to reduce the cost.
Yes.
Some of the primary ways to reduce the cost of our product is teaching our yeast how to make more protein per unit. So we are constantly tinkering around with 5,000 genes that are in our yeast, tuning them up and down and trading things in and out to figure out what is going to be the most successful intervention or change to the yeast strain that's going to produce the highest yield.
Yes. And really what the yeast are functioning as, mini factories, really, to produce this protein. And you want as much of this protein as possible.
Exactly. So we put our yeast cells into a tank called a bioreactor. In the bioreactor, we feed our yeast cells food and different components, air and oxygen, to make sure that the yeast has the optimal conditions to grow and spit as much protein out of the cell as possible. So this is our Afera, Actoferrin, the pink powder. It's pink because it binds iron. So it gets this light pink color, but when it's added to other products, it kind of dissolves in. You don't notice the color, but this is what it looks like when it comes out. If we move down our hallway over here, we have our bioprocessing room. And in here, we've got bioreactors that are fermenting our yeast. So we have many different fermenters here at small scale. And this directly correlates to our production at larger tank sizes with our manufacturing partners. So we've got 1, 2, 3, 10-liter fermenters here. At commercial scale, we have 50,000 liter tanks.
Wow.
So we've miniaturized the process here and we do a lot of testing to see what process conditions are going to be exactly perfect to provide what the yeast need to be as productive as possible.
Great.
That's the bioprocess room.
And what do we have next up?
Moving up, we've made the protein at this point. We now take it and we have to see how it works in food. So we have a food science lab. Most people say, don't eat your experiments, but that's exactly what we do here. We take our lactoferrin, our Afera, and we test it to see how does it work in beverages, bars, gummies, infant formula. All of the application work is done here.
Smells like milk in here.
Smells like milk. We have a spray dryer in there. So we test drying different products, including formula, sports drinks, protein powders. So it smells very sweet.
No, so it's fabulous. It's really... And in that fridge, you have the test products, right? Whether it's
all
a
different kinds of
it's a
products.
gum, and all of that. And so who are the human testers who are actually tasting it?
We taste it.
You taste it?
I've tasted so many different products here. We are constantly working with our customers who are making different products and showing them how they can use Afera in a new product they want to launch. So we've made drink mixes and protein powders and gummies and all kinds of things, and all of that tinkering, we'll taste it here ourselves.
Yeah, that's great. That's excellent. Right. So what do we have now behind here?
Behind us, we have several different, very specific labs for our yeast work, our mammalian tissue work, and we've got a mass spec. So behind us, we have dedicated labs for our yeast engineering, how we engineer our yeast. That's our yeast tissue culture room. We have a mammalian tissue culture room where when our protein is ready, we take the protein and we test its functional properties in mammalian cells, so in primary cell lines and human cell lines to look at the efficacy. And then we have a mass spec room dedicated to all of our protein analytics.